Through glycolysis, one glucose molecule is broken down to make 2 NADH, 2 net ATP, and 2 pyruvate. The 2 pyruvate produced after glycolysis enters the mitochondria through pyruvate oxidation, also known as intermediate reactions. The loss of electrons from the pyruvate joins with NAD+ to make NADH, a high electron carrier. In addition, carbon dioxide is lost and allows for the 3-carbon molecule pyruvate to turn into a 2-carbon molecule known as acetyl-CoA. Acetyl-CoA will then bind with a 4-carbon molecule known as oxaloacetate, which would then start the Krebs cycle and allow for a 6-carbon molecule to be produced, also known as citrate. Citrate will then lose a CO2 and also allow for an NADH to be produced when NAD+ gains electrons. This would happen twice and allow for 2 NADHs to be produced and 2 CO2s to be released. Then adenosine diphosphate and inorganic phosphate would then bind together to make adenosine triphosphate, also known as ATP, the final energy product. Also, another high electron carrier known as FADH2 was produced when FAD+ gains electrons, and finally another NADH is produced when NAD+ gains electrons, and this process is repeated twice to make a net of 6 NADH, 2 FADH2s, 2 ATPs, and 4 CO2s released. And this is produced for each glucose molecule introduced into the Krebs cycle, also known as the citric acid cycle.
NADH and FADH2 are high-energy electron carriers that make their way across the inner membrane of the mitochondria towards O2. O2 is extremely electronegative and it attracts the high-energy electron carriers. Special electron carrier proteins take NADH and FADH2 towards O2. They release electrons and protons out of the matrix and become NAD+ and FAD+. In order to get back into the matrix, the protons use the enzyme ATP synthase and then bond with ADP to form ATP. When the electron carriers make their way to the O2, they bind with it to become H2O. Just one glucose molecule can make 30 to 32 ATP.
Alternatively, ATP can be produced through the metabolic process known as fermentation. Fermentation occurs when there is an insufficient amount of oxygen to be used for the electron transport chain. There are 2 types of fermentation: alcohol fermentation, which occurs in yeasts and bacteria, and lactic acid fermentation, which takes place in muscle cells. Both forms of fermentation start with glycolysis. After the glucose molecule has been broken down into 2-pyruvate in alcohol fermentation, it is then oxidized into 2 ethyl alcohols. However, in lactic acid fermentation, the 2 pyruvates become 2 lactic acid molecules. In both forms of fermentation, one glucose molecule will yield 2 ATPs.